welcome. Thank you.
Okay. So moving on, our next third presenter today is Dr. Rebecca Weed. Rebecca completed her PhD in molecular plant sciences at Washington State University. Using a metabolomic approach, her doctoral work focused on understanding allelopathic interactions in invasive plants and how fungal pathogens can modulate the plant host's metabolome to escape detection. Following that, she moved into her current role as a research scholar at North Carolina State University's Analytical Core Facility, METRIC, where she has mostly worked on developing methods to quantify per and polyfluoroalkyl substances, or PFAS, from either water or biological matrices. Recently, she has developed a method for untargeted PFAS analysis and has worked on developing other quantitative methods for a variety of small molecules. Thanks so much for joining us today, Rebecca. You can start your presentation now.
Thank you so much, Ben. Good morning, and I'm excited to present to everyone today. Go ahead and share my screen. All right. So today I'm going to present to you a talk about dirty water, clean data, how we measure and detect PFAS using stable isotope standards. First off, PFAS has already been mentioned a few times, but I just wanted to give a quick definition of what it actually is. PFAS, or per and polyfluoroalkyl substances, are essentially compounds that contain at least one fully fluorinated methyl or methylene group, as shown by the structures on the right. There have been over 9,000 PFAS identified. That was really cool, Dr. Nakayami, that you got to work with Mark Schreiner back in the original PFAS days. So in North Carolina, we have one chemical manufacturer plant, or we have many chemical manufacturer plants, but one of notoriety. It was originally called DuPont. They rebranded maybe like 10 years ago to be called Chemours. And their main fluorochemical chemistries are Nafion, Teflon, and Viton. And you can find these chemistries in pretty much every industry that you could think of. Nafion is found in energy, transportation, chemical processing. Teflon, you probably know from your nonstick pans, textile finishes. It coats like fishing line so it doesn't tangle. And Viton is a fluorochemical rubber, and it's found in the automotive, aerospace, and oil and gas industries. And the main challenge with most fluorochemical chemistry is not the production of these products. It's actually the all of the different PFAS molecules that are needed in order to produce these chemistries, and they're called byproducts of the chemical production. These are ultimately released as waste products into our local waterways, and additionally, all of these different products, whether it be your fishing line, your cookware, etc., are eventually going to be sent to the landfill, and where they're going to degrade and release these different breakdown products of PFAS into our waterways, the groundwater, etc. In North Carolina, we have a lot of PFAS contamination, unfortunately. We have many different locations. Shown here is a map of North Carolina on the bottom, and then highlighted in red is the Cape Fear River system. This supplies majority of the water to drinking water to most North Carolina residents. And along this waterway system, we have different industries such as Elevate Textiles in Burlington. We have multiple airports, RDU, and many different military bases such as Fort Bragg, Camp Lejeune, and the biggest ammunition terminal in the world, which is the Military Ocean Terminal in Sunny Point, which is just south of Wilmington, North Carolina. And all of these contribute different types of PFAS into our waterway system that eventually dumps into the ocean in Wilmington, North Carolina. Due to such heavy PFAS contamination, we are the site of a Superfund for PFAS contamination. a few years ago, North Carolina State University won a Superfund grant where we were then able to set up a research center focused on PFAS. My team at Metric was responsible for doing all the analytical chemistry for the center. We did all the mass spectrometry and quantitation of PFAS for this grant. Through this, we've had many different projects on PFAS, but the one I wanted to focus on today was actually just published this year in ES&T, and it focused on the detection and quantitation of PFAS in North Carolina sea foam and the corresponding seawater. So why do we care about foam? Well, first off, if you've ever looked at the ingredient list on your shampoo bottle while you're taking a shower, on the back of it, you'll see an ingredient called laureth sulfate, and it is a very similar structure to lauric acid. And lauric acid happens to be a direct corollary to a very common PFAS molecule, PFD-OA. In fact, if you just pop off the hydrogens and replace them with a fluorine, you now have a PFAS molecule. All of these molecules have a hydrophilic head and a hydrophobic carbon tail. So when they're in water with the presence of agitation, these molecules are going to stack in a way that they form a 3D structure, a sphere, aka a bubble. And all of those bubbles coagulate, and then you now have foam. And as you know, if you've ever taken a bath, those lovely bubbles and foam eventually dissipate in a few hours to maybe a couple days. However, foam that has been augmented with PFAS molecules has the environmental half-life of forever. There is no, nature has no solution for breaking down this carbon-fluorine bond. Hence the reason why these compounds are called forever chemicals. So we all know that sea foam itself is natural. It is generated by the agitation of water containing high levels of dissolved organic matter. That organic matter can come from many different things, such as like algae bloom breakdowns, etc. We've had a lot of people ask us, how do you tell the difference between sea foam that is natural versus PFAS based? Anecdotally, people have said that natural foam is off-white, has an earthy smell to it, and that PFAS foam is bright white, sticky, kind of like shaving cream. And unfortunately, those are just anecdotes. There is absolutely no way to tell the difference between a natural sea foam versus foam that has been contaminated with PFAS. We are not the first group to look at foam, but we are the first to look at sea foams. Up in the Great Lakes, there are two studies that were looking at foams and freshwater locations, and those foam events were mostly the result of some industry, but mostly landfill leachate effects. The three locations they looked at were Glen Lake near Traverse City, Michigan, Muskegon Lake near Grand Rapids, and then Starkweather Creek near Madison, Wisconsin. And these studies found PFAS levels in the foams to be as much as 7,000 times the levels of PFAS in the surrounding water. Due to those results, the Michigan Department of Health and Human Services and the corresponding Wisconsin Department of Health Services both recommend for everyone, people, pets, etc., anywhere that there's known PFAS contamination, to just basically avoid the foams. So back to our project. A graduate student, Drake Phelps at the time, was working closely with a community partner, Emily Donovan, who is the co-founder of Clean Cape Fear, a volunteer grassroots community group focused on the conservation and cleanup of PFAS. He originally brought this project to us. Emily actually lives in Wilmington, North Carolina, and she had noticed an increase in the abundance and occurrence of these foam events along the beaches where she, played and lived with her children. And she thought maybe there was a relationship with all of the PFAS pollution in the area, and so asked our group if we could investigate this for her. So what we did was we sent collection kits to Emily, and she kept these in the trunk of her car. And anytime she saw a large foam event, she'd pull over and she would collect bulk water that was either directly underneath or right next to the foam in the water. And she would also collect the foam. And this took about a year to get enough collection samples. The foam events are very sporadic. It really depends on the weather, the tide, what's accumulating from upstream sources, etc. Once we felt that she had collected enough different samples, all of them were shipped to our lab, and then we um, we analyzed them via mass spectrometry. And I just wanted to point out again, just north of Wilmington, there's the airport, two different airports, and then again, Matsu, which is the ammunition terminal. And due to the high risk of what is being stored at that ammunition terminal, they have very strict procedures for safety and the use of firefighting foams, which historically are very full of PFAS. So, how do we process the samples? We spiked the samples with an internal standard mix and then extracted them via SPE using a wax cartridge. The extraction method was a slightly modified version of the EPA 537 and 533 if you're interested. We then dried the samples to completion and then reconstituted using a 50-50 mixture of methanol and water. This was analyzed then on one of our mass spec platforms. We used a Thermo Scientific Vanquish LC using a Kinetics F5 column for analytical column, and then a Thermo Scientific Orbitrap Exploris 240. The exact method details are found here in this paper if you are interested. Just a quick aside, I want to just highlight the importance of stable isotopes and especially for quantitation. Ionization is not always consistent and can drift over time due to several reasons. The biggest being your source can get dirty due to repeat injections of complicated matrices, and then your actual matrix can change. Biology is not consistent. So we can solve this by normalizing our calibration curve of our light to heavy peak areas. And shout out to CIL. We have 3 separate mixtures that were born out of a collaboration with Cambridge Isotopes. We have 2 light mixtures and 1 heavy. And we were very excited to collaborate with CIL on this. They were able to produce an internal standard mixture for us that had many more heavy atoms compared to other competitor vendors, which is always great, when you're doing mass spectrometry. And we are thinking about potentially updating these mixtures a little bit just to stay up to date on the current PFAS research. Let's look at the results. Okay, so there's a lot going on with these graphs, but what I want you to focus on is that they are separated by chemical class, which is based on the PFAS functional groups, whether or not you're carboxylic acid and ether, etc. And all of these results are for the bulk seawater. And the main thing to take away from this is that one is PFAS in every single sample. And then if you look at plot H, this is a plot of all of the combined PFAS. It is in logarithmic scale, so every tick mark is 10 times the previous tick mark. And overall, the levels for the bulk seawater are not dangerously high. It's higher than perhaps what you'd want in drinking water, but not crazy. Honing in on 2 of the graphs, these compounds are generally accepted as Chemours-specific compounds due to the fluorochemistry that they perform at that plant. This company you really wouldn't find these anywhere else in the country. You might be able to find them in China or in some Asian areas. Something generic like PFOS would be incredibly hard to track because there are a myriad of different sources for that compound. But we know that these are specific to the fluorochemistry done in this location. And so, yeah. All right, so let's look at the sea foam concentrations. Again, we have the same organization based on PFAS class, same, uh, yeah, or within each PFAS class, we have the different colors representing the different PFAS compounds. But what I want you to look closely at is the y-axis. For the bulk water concentrations, we were in the tens to 100s of nanograms per liter. And we are now in the 10,000, 100,000, or even in the million nanogram per liter concentration. In fact, one sample 10, had PFOS concentrations greater than 8 million nanograms per liter. And again, um, every single sample had a lot more PFAS of all types. Um, these levels are the highest ever reported for water-based environmental foams, exceeding the previous values recorded in Michigan and Wisconsin. And then again, looking at the more specific compounds, some of these are in the order of 30 to 40,000 nanograms per liter for some of the ether carboxylic acids, and then on the order of 10 to 15,000 for the ether sulfonic acids. And then looking at the H plot, which again, logarithmic scale, a lot of the samples are approaching 10 to 6, which is 1 million nanograms of PFAS overall. And if there's nothing else that you take away from this talk, this is what I want you to take away, is that these are just the PFAS that we are quantitating and that we have analytical standards for. Our method has around 50 different compounds. And if you remember at the very beginning of my talk, we know there's over 9,000 PFAS compounds out there. So this is just a drop in the bucket of what we are actually quantitating. Yeah. So not only did we do a quantitation study, we also did a untargeted method to identify potential unknown PFAS. This consisted of two different experiments. Experiment one was an untargeted suspect screening. How this worked was that, or how this method worked was that we have a full scan event that utilized a suspect screening list that we obtained from the EPA CompTox website. Database, and that allowed us to prioritize the selection of PFAS masses. If any of those masses were detected, they were moved to a dynamic exclusion list, so they wouldn't be selected for again. The most abundant masses detected were sent for fragmentation, and then the cycle was repeated every 5 scans. The major limitation with this is that, again, only the most abundant precursor masses are sent for fragmentation. Anything that's in low abundance is going to be missed, and we need that fragmentation info for compound identification. So we performed a second experiment using a thermo-specific workflow called AcquireX. Essentially, how this method works is that you have your a blank sample, um, and you do an injection of that. You create an exclusion list based on that because you don't want to really care about your background matrix molecules. You then have a pooled sample of all of your unknown samples, and you do a full scan injection of that to create an inclusion list, and then do iterative injections of that pooled sample. Each injection, the most abundant masses within that list are sent off for fragmentation. Once they're sent off, they're moved to that exclusion list so you don't reanalyze them. And then you just keep moving down that inclusion list to try to get as many low abundant compounds as possible. All of this data was then analyzed in ThermoSoftware Compound Discover. And we use the latest PFAS workflow for that with all the additional add-on scripts, including like the Duke Spectral Library. And if you are interested in the specific details for the data analysis, please view the supplemental section of our paper. After filtering all of those results, we had 516 features remaining. And as Dr. Kovaci already presented this, this is the confidence scale that was published by Emma Szymanski back in 2014. It has since, this is her original scheme, but since then, in collaboration with Joseph Charbonnet, an updated version of this has been published for PFAS-specific research. We followed this our targeted results were then sorted based on these confidence levels. Essentially, if you have an analytical standard that you've run on your instrument and you compare it to your unknown result and it matches, you have a level 1 identification. The remaining results are sorted based on the amount of information that you have available. Following this type of sorting scheme, we had 50 unique features that were identified at a level 2 or 3 confidence level. I don't have time, unfortunately, to present all of those to you, so I'm going to just highlight one of them. And the compound that I chose from our manuscript was perfluorohexane sulfonic acid, or PFHX-SI, and it is a level 2A, so as high as you can get without an analytical standard. This compound had a matching isotopic pattern with its chemical predicted formula and a mass error of 0.5 ppm. Additionally, the retention time of this analyte aligned with the estimates based on our standards. The product ion spectrum collected from a sample from 10 corresponded to this identification within the Duke spectral library with the 3 most abundant ions that are shown here, 318, 168, and 118. Finally, using structure logic and comparison to results within the literature, we found additional evidence to support our identification via via the manuscript published by Bao et al. In 2023. They found this compound in soils from that were nearby a former fluorochemical plant in China. And again, they had the same matching product ions. A separate paper published by Wang et al. Reported this compound in AFFF contaminated groundwater in Pennsylvania. AFFF is an acronym that is associated with firefighting foams. Due to the presence of this compound, we feel that it may indicate the presence of zwitterions or sulfonamides that are from a firefighting foam origin in the sea foam. And we think this could be related to proximal facilities such as local airports and the Matsu ammunition terminal. So summary take-home message. PFAS are homophilic or fluorophilic. They tend to prefer interactions with other PFAS more than interactions with water or oils. Therefore, once the PFAS is in the foam, they're very unlikely to migrate back into the water. Water-based foams are potentially significant sources of PFAS exposure. Some sea foams collected along North Carolina beaches contained extremely high levels of PFAS. Some of the highest PFAS observed are known Comores-specific compounds. And finally, Emily Donovan, through Clean Cape Fear, is using these results to impact environmental and health policies at the local and federal level. A big shout out to the team. This would not have been possible with everyone shown here. Thanks so much.
All right. Thanks very much, Rebecca, for that great presentation. I think we all know North Carolina is a hotbed for PFAS interest, but it's really interesting to see your perspective on the coastal contamination and especially the levels that you've identified. That's pretty interesting to say the least. In the interest of time, again, I think that we're going to move right along. We do have one question in the Q&A section. If you could maybe answer that back, type an answer for that, that would be great. We appreciate
Absolutely.
it.
Thanks.
So, yeah.